Okay, so let's go ahead and pick up where we left off. So working through the four main areas of the brain. So we left off at cerebellum. So we already talked a little bit about the cerebellum. So this is just kind of like an anatomical view of it. And so a lot of you guys recall seeing this when you're working on the sheep's brain. So this is a oops, okay. This is a sagittal view of the brain. So for instance, you've got your brainstem here, medulla, pons, midbrain, and then of course your cerebellum here. And you can see this tree-like structure, the white matter, the arbor vitae, and then the folia, the little folds, basically leaf-like structures that kind of fold around that arbor vitae structure. Another thing that's worth mentioning that a lot of you guys noticed on your sheep brains is your fourth ventricle, which basically is kind of wedged in between the cerebellum and the brainstem, and then your cerebral aqueduct, which connects the third ventricle to the fourth. And several of you guys saw that on your sheep brains. And so when you are taking a look at the structure of this area, so like for instance, if you were to cut a little piece of this out and blow it all up, you can see your different types of neuron cells. So you can see your Purkinje cells. Your Purkinje cells are important because they're like little twig-like fibers that permeate all the nooks and crannies of tissue. This is kind of like an extension of a neuron so that you can really get into those deep corners of tissue. You see a lot of Purkinje fibers, for instance, in your cardiac muscle, so that every single cardiac cell can have communication to the nervous system. And then you have a couple of different types of cells. So you see a Purkinje cell, and then you see your stellate cell, which is kind of like a little satellite, like a little outpost of like twig-like neural fibers that also helps to extend the reach of the neural communication beyond just the core, the core components. And then you see what's called a basket cell. Mostly this is because when anatomists looked at this, they said it kind of forms a basket-looking structure. But you can see it's variations on the same theme, where you have just varying degrees of sort of like these little fiber like extensions that are all kind of serving similar purposes in the sense that they're trying to permeate all the tissue. And you can kind of see a slightly different variation here where you see a cell body attached to a big swollen area. It almost looks like a spider's abdomen. That's a Golgi cell, and you can see you have these little dendritic-like structures that are kind of permeating throughout that tissue. So you can see lots of nervous system, lots of nervous innervation in that particular area. This is one of the reasons why we say that the cerebellum has more neurons than the average region of the brain. And this is kind of what it looks like when it's dissected out. So you can see you have the two main structures. The hemisphere, your lateral hemisphere, is kind of broken up into lobes. And I mean, some of it, you honestly can't see a lot of this because when you take a look at the cerebellum, it kind of all looks the same. So you kind of have to really sort of really focus in and concentrate on it. But there are lobe-like structures to it, like subdivisions, if you will, within these larger sections. For instance, there is a little gap that you can separate between anterior and posterior lobe. That's the primary fissure. And then that kind of separates this out into sort of sections and layers. And so you can see that structure in the cerebellum. And so that's just kind of basic, the basic anatomy and the structure. But, you know, from our models, it pretty much all looks uniform. They don't really do a very good job on our models of sort of get to that fine level of differentiation. So the next major region of the brain, so we've already covered two, right? So we've got the brainstem and we've got the cerebellum. Now it's time to move into the diencephalon. Of course, we're kind of slowly moving from the base of the brain on up. And the diencephalon is going to be the area between the brainstem and the cerebrum. And this is where you have a lot of those, thalamus, the thalamus family, right? So you have your thalamus proper, your subthalamus, your epithalamus, and your hypothalamus. So these are all kind of part of the same region that essentially is composed of this the main section between the cerebrum, which is up here, and then of course the brainstem, which is just underneath. So it's kind of like down in this region on down. So a couple of things, first of all, to point out in terms of the thalamus. The thalamus is important. We'll take a closer look at what each of these pieces are doing and their importance in terms of the brain. But a lot of you recall when you did your sagittal view of the brain, you saw this kind of ram's horn looking structure, which is the corpus callosum. And so this is an important structure, an important landmark in that sagittal view. And then beneath it was the fornix, and then the open space is one of the ventricles. So the lateral ventricles, there's 4 ventricles. There's first and second, which are both lateral, third, which is underneath the laterals, and then the 4th ventricle, which is down by the cerebellum. Another structure that you'll see just below that corpus callosum and fornix area, that's where you're going to find your thalamus. Now, in your sagittal cross sections of your sheet brain, that just looks like a nondescript sheet of white tissue. And so sometimes it's easy to see, sometimes it's not. But if you were to take a closer look and kind of really get into kind of a deep dissection, a really focused dissection of the thalamus, what you're going to start to find is basically the overall bolus, that the majority of what you're seeing in that particular area is thalamus. And then you oftentimes will see this little bean-shaped structure that sort of is right in the middle. And that's what's called the interthalamic adhesion that connects basically the two halves of the thalamus together. And so that typically will form your thalamus. And then your epithalamus has a couple of major pieces to it. The habenula, which is kind of like in the back of the thalamus, and just below that, the pineal gland, which is this kind of like pine nut shaped structure that's right behind and just above the superior colliculus. And so sometimes when you kind of bend the brainstem back, you can actually see just a little itty bitty kind of a ball shaped structure that is the pineal gland. I was looking for those in the intact sheet brains. I didn't really see a good example of one, but I will continue looking. And if I find one, I'll show you guys during lab. The pineal gland is important because it secretes melatonin, which is essentially kind of our it's kind of like our sleep aid. So melatonin makes us drowsy, and that kind of helps us to fall asleep. And then below the epithalamus and the thalamus is going to be the subthalamus, which is kind of like this region that sort of is sandwiched between the thalamus and probably the most important. Well, the thalamus is important, but the hypothalamus is really important because there's a lot going on in the hypothalamus. This actually forms a region of the brain that is kind of the main controller for a lot of really critical sorts of things. So the hypothalamus is really an important and critical kind of extension of this diencephalon region. Because ultimately, when you take a look at it, it forms kind of the top of an entire system, which is a critical and crucial system, your endocrine system. And so the hypothalamus is really important. As a matter of fact, in AMP2, you spend a lot of time talking about the hypothalamus because it is part of the visceral control of a lot of your organ systems. Okay. So they kind of color coded it here. So the anterolateral view of the thalamus, so you can see it's not just a sagittal view. So you can see basically it's 2 halves, and the interthalamic adhesion, it basically kind of connects the 2 halves together. It's almost like an isthmus, like a little bridge area that will connect these 2 halves of the thalamus together. And within the thalamus, you'll see these color coded structures. These are the different nuclei that you're going to find in the thalamus itself, right? For instance, you'll find what's called the anterior nucleus, the ventral anterior nucleus. A lot of these are named after their position. You'll see the ventral lateral, the ventral posterior, the lateral geniculate. I always get that one. I always want to say genticulate. It's geniculate nucleus. So this is one that we oftentimes will see a lot of literature on. The pulvinar, the lateral dorsal, and the lateral posterior nucleus. So you'll see there's a lot of different nuclei that are involved here, and they all have slightly different jobs. They're separated from each other, not just by the interthalamic adhesion, but also by the what's called the medial nucleus. So that's kind of like the I guess it's not really a separate. It's not structural in nature. It's a nucleus. It's doing processing and stuff, but it kind of sort of cuts between the two halves, sort of like a little intervening line that sort of demarcates where the two halves of the thalamus meet in that particular area. Now, the hypothalamus has also a lot of nuclei. And so these are oftentimes named after both position and function. So when the anatomists were taking a look at these, they were largely looking at positional types of things. It's because, generally speaking, we were discovering structures and we could describe where they were, but we didn't really know what they did. And that's the reason why you have a lot of these sort of positional terms. Like, for instance, one area, they're not all positional, but one is the preoptic area. So before the optics. So that's basically kind of positional, telling you where it is in relation to the optic nerve. You can see the pituitary, remember, is right next to and behind the optic chiasma, which is where the optic nerve comes in. So that's what they call that. A couple of really big ones and important ones, especially for AMP2, is the supraoptic nucleus. So supra means above the optic, so you can see the optic chiasma. And the suprachiasmatic nucleus. So this is like right at the tip of where the backside of the optic chiasm would reside. And so we hear a lot of work on these two nuclei, especially the suprachiasmatic nucleus. You also have the anterior nucleus, which is this blue guy right here, the ventromedial nucleus, which is the red guy, the dorsomedial nucleus, and the lateral area, as well as the posterior and now the mammillary body. Many of you guys remember seeing the mammillary body. So some of you will notice that a couple of you guys saw this in your brain models, right? So you'd see the pituitary and the infundibulum, and then right behind and at the base of the infundibulum would be these little white spots, and those were the mammillary bodies. And sometimes you can see the mammillary bodies in the sheep brain as well. Just underneath the ventromedial nucleus is the arcuate nucleus. And the other big one that we hear a lot about is the purple one. So this is going to be the paraventricular nucleus. So para basically means beside, the ventricle. A nucleus, again, it's a positional term. So the ones that are bolded are typically the ones that we hear the most about. It's the ones that we know the most about, and it's the one that there's a lot of research about. So there's a lot of nuclei in these areas. So let's kind of get a little sense of what maybe some of these areas do so that we can actually kind of round out our knowledge of the thalamus and kind of get a sense of what's kind of going on here. So lateral portions, which are that's basically the thalamus. So they're interconnected by the interthalamic adhesion or the intermediate mass. Sometimes you'll hear those two terms are synonymous. And it's surrounded, this particular structure is surrounded by the third ventricle. So you don't oftentimes see the third ventricle in the sagittal view. You can see it a little bit better in the coronal view. But this kind of surrounds that intermediate mass of the interthalamic adhesion. What the thalamus acts to do is basically it's a relay center. So typically sensory information are essentially moving through the thalamus and they're projecting to the cerebrum, to the various areas of the cerebrum, and the different nuclei are designed to handle the different types of sensory inputs. For instance, the medial geniculate is going to be handling auditory information. The lateral geniculate is going to be handling visual information, sensory information that's coming in. The ventral posterior will handle pain, among other types of sensory information. Motor function is being dealt with by the ventral anterior and the ventrolateral nuclei. So this is kind of where some of your motor signals are coming through. Mood, right? Mood modification. That sounds terrible, doesn't it? This is the anterior and medial nuclei, right? So these guys are basically taking in the different kinds of communications. I don't want to call it sensory information because it can also be effector communications as well. They could be going out as opposed to coming in. But these guys are connected to your limbic system, and your limbic system is a region in your brain that's responsible for your emotions. And so this particular area, the anterior and the medial nuclei of the thalamus, are the ones that are kind of regulating and sort of handling all the incoming messages and the outgoing messages associated with that particular area. In terms of, and so that's one particular piece, right? So you're basically taking stuff in, taking stuff out, but you also have an extra nucleus, the lateral dorsal nucleus, that's also involved in the regulation of emotion. A couple of other nuclei that you see in the thalamus, so the lateral posterior and the pulvinar, these guys are essentially working on sensory integration. So this is a really, really good slide for the thalamus. This is basically, you can title this slide, "What Stuff in the Thalamus Does." So if you want to know what the stuff in the thalamus does, this is the slide to go to. Okay, so that's the thalamus. Let's move into the subthalamus, right? That's that region that's immediately below the thalamus. And so this works along with, as part of the basal nuclei, the subcortical, which basically means subcortex, basal nuclei, and the basal nuclei is involved in motor function and control of motor function. So a lot of these nuclei, the subthalamic nuclei, will be taking in ascending and descending nerve tracts, but it's basically handling all of the motor function and motor control. So you notice already that we've already talked a lot about motor control, right? I mean, we had nuclei in the midbrain that were involved in motor control. We have the cerebellum, which is involved in motor control. We had the cerebral peduncles, we had the cerebellar peduncles, which were connecting to these regions so that these different areas of motor control can communicate with each other. And now in this diencephalon area, in the subthalamus, we have yet even more motor control. So these particular specific areas that you're going to see, like for instance, the habenula, this is going to be a controller of a lot of different things, but this is going to help you with your what's called motivational control of behavior. And so this is typically tied to your limbic system, right, which is where your emotions are coming from, your basal nuclei, which is where your motor control is coming from, and other parts of your brain that is involved in behavior, and like your reward system and things of that nature. So this is an area that is in touch with a couple of things. So if you take all these together, you're talking about your emotions, your motor control, your reward system. And so a lot of this has to do with impulse control, right? So impulse control is kind of like when your emotions are having sort of unregulated activity. So basically you're not thinking about what you're going to do, you're just doing it. And the act of doing that gives you immediate gratification, which then ultimately causes you to have this kind of reward, right? So that's kind of where some of these addictive behaviors come from and things of that nature. So it's kind of complex, right? Because there's a lot of really complicated areas that are involved in this particular section. And so that's the reason why, you know, we're really studying a lot of this, and we just kind of scratch the surface on it. The pineal gland is certainly considerably more simple, relatively speaking, because this one is in charge of the sleep-wake cycle and your circadian rhythms. And it's the secretor of melatonin. Like I said, it's basically going to make you drowsy. It's also light sensitive. So when the lights go out, your pineal gland starts to stimulate and starts to create more melatonin, which makes you drowsy, which is the reason why we tend to get a little sleepy when the lights go off. So the next section is going to be the hypothalamus. So we covered thalamus, we covered subthalamus, which is motor control, epithalamus, which has kind of your impulse control, your emotion, basal nuclei, and pineal gland. And now we have the hypothalamus. And so this is the bottommost part. So this is like the bottom level of this thalamus region, this thalamic region. And there's several different things going on here, obviously, because we mentioned them a little bit. So the most inferior portion is going to be, the mammillary bodies, which we kind of pointed out right behind the infundibulum. These guys basically are going to be responsible for olfactory reflexes. So that's your sense of smell. And they're also going to be tied to emotional responses to odors, right? So if you have an emotional response, what that means is it's essentially integrated with the limbic system. So if you're paying attention, you might be catching a theme here that we're talking about a lot of different nuclei in a lot of different areas, but these things are not working in isolation. They're all integrated and connected to each other. And there's a couple of these areas that keep coming up, right? So the limbic system is a big one because that's one of the things that is one of the most advanced features of our brain. And so when you have olfactory reflexes and this sort of memory tied to odors in charge of your mammillary or being driven by your mammillary bodies, this allows you to be able to reinforce also higher order learning, which is cerebral cortex. So a lot of times this will help us to respond to odors, but then that response to odors can then be communicated to the other parts of the brain so that we can remember our response to this particular odor. In this particular case, a reflexive response to the odor can be, say, for instance, a general sort of perception of disgust. You know, maybe if something stinks and it's unpleasant, right? That would be an emotional response to an odor that would be involving the mammillary bodies. The infundibulum, which is just in front of the mammillary bodies, is essentially a structural stalk that connects the hypothalamus to the pituitary gland. And so in this stalk, you also have different types of neurons that are going from the hypothalamus into the pituitary gland. But a couple of important nuclei in the hypothalamus, at least for the purpose of the pituitary, are the paraventricular nucleus and the supraoptic. Because together, these will secrete hormones that are part of the endocrine system that will often have broad effects in the body. So just like the nervous system is regulating the body for short-term responses, the endocrine system regulates the body for long-term responses. And so the paraventricular nucleus and the supraoptic nucleus are actually responsible for secreting and making a couple of hormones that the pituitary will then release. And so these guys are important. The hypothalamus will be taking in a lot more information. So there's a lot of sensory information coming into the hypothalamus. So there's a lot of homeostatic information. So a lot of homeostatic information comes from the body to the hypothalamus. And so it kind of sorts through that. For instance, you have a lot of these inputs from internal organs. That's those homeostatic types of inputs. Your taste receptors, so your sense of taste will go to your hypothalamus. Your limbic system will go to your hypothalamus. Nipples, external genitalia, the eyes. So there's a lot of stuff coming into the hypothalamus. And it's basically sorting through all these different inputs. So when you're taking a look at this thalamic region in the diencephalon, what you're looking at is basically an area that's really responsible for just fielding just tons of incoming information from the body, and it's being divided up. The labor of that in that sort of reception of receiving all of that information is being divided up by into these different nuclei, and each of these different nuclei are specialists for those particular incoming types of sensations. So one thing that we see in the hypothalamus is that it is tied to the autonomic nervous system. So efferent, or those are exiting fibers, will go to the brainstem, to the spinal cord, and then those will. You also have fibers that will go from the hypothalamus to the pituitary. So these are different regions. So there's one set of fibers that will go from the hypothalamus to the brainstem to the spinal cord. A second set will basically communicate from the hypothalamus to the pituitary gland. And then, of course, you have cranial nerves, which are also important in this particular area, that will control different types of things. Shivering, for instance, which is an important one, because your hypothalamus is essentially your thermostat. So when you're controlling body temperature, you're controlling that body temperature, and that's being controlled by your hypothalamus. So if you're increasing your body temperature because you got a fever, that's your hypothalamus doing that. If your core body temperature starts to drop and you need to start generating heat, your hypothalamus is the one that induces the shivering of your muscles to create more heat. So you'll have a lot of different things, for instance, happening in this region. So different areas of hypothalamus will add to mood regulation. Your emotions can be regulated by your hypothalamus, sexual pleasure, your sense of satisfaction, satiety, rage, fear. So all these emotions that really kind of set us apart, a lot of these are being handled by the hypothalamus in conjunction with the limbic system. So you can see that there's, I mean, we have, our emotions are such a mess that we have like really, really enormous in multiple areas of our brain that has to handle and deal with emotions and sort of regulate them and connect them properly so that they can function in a constructive way. So another one that you hear a lot about, and there's a lot of research on this one, is the suprachiasmatic nucleus. And the reason why there's a lot of research on this one is because along with the pineal gland, this one is kind of in charge of our light-dark circadian rhythms. What does that mean? Basically, a circadian rhythm is like, are you active at night or during the day? And it doesn't mean, are you a night person or you're a morning person? That's not what it means. Every organism has a circadian hardwired into them, a circadian rhythm hardwired into them. So some animals are nocturnal, which means they are awake and active at night, and they typically sleep during the day. Other animals are diurnal, which basically means they are active and awake during the day, and then they are at rest at night. Those typically are fairly hardwired into the nervous system. And so this is one of the reasons why doing night shifts is typically difficult, and why it tends to create a lot of problems for individuals, and why insomnia creates a lot of problems for individuals physiologically, because it creates a lot of negative wear and tear on the body, because we are hardwired to be diurnal. Doesn't matter if we work night shift, your body doesn't care. Your body still says, listen, I'm diurnal, and so you can work at night and try to sleep during the day, but it's not going to work as well long term. And so those are coming through that suprachiasmatic nucleus. So this is kind of like just sort of a general overview of some of the functions of the hypothalamus. I'm not going to go through this. I think we kind of went through quite a bit on the hypothalamus, and I think it's probably sufficient to give you guys a pretty good start for AMP2. So now what we want to do is we want to basically move into our last region of the brain, the cerebrum, right? So just like our limbic system is one of the innovations that we have in our brain, the cerebrum is also one of the innovations we have in our brain. The ability to have higher order thinking, reason, logic, awareness, things of that nature. So the first thing we need to understand is the structure of that particular area of the cerebrum. So this is going to be a big piece of our brain, the largest portion of our brain. It's divided into two halves, kind of like the south thalamus was. So you have right and left hemispheres, and you also have an inner and an outer region, right? So the cortex, the cerebral cortex, is essentially the outer surface. And the idea is when you see these two terms, cortex versus medulla, this is a generic descriptor, right? So cortex typically means surface. So you have a cortical region of your kidneys, and the medulla basically means the inner core. So if you have a medullary area, it's in the middle core surrounded by a cortex, the outer layer. And so you have adrenal cortex, adrenal medulla, you have a kidney cortex and a kidney medulla, you have a cerebral cortex, and of course you have your medulla, which is the medulla oblongata. And so that medulla basically means it's the inner core piece. Now the brain itself, or the cerebrum, is basically constructed of folds and grooves. So the folds are kind of called the gyri. So these are kind of like the ridges of the fold. So if you take a look at a brain edge on, you'll see these kind of like little folds and grooves and folds and grooves. And so a gyrus is basically the upper portion of that fold. The sulcus is the depression. So the little groove or the crevice is going to be the sulcus. Now we use different gyri and different sulci as sort of landmarks in the brain. For instance, a couple of major ones, and a deep sulcus is a fissure. So a fissure is defined as a deep sulcus. So for instance, one thing we notice in the brain as a landmark is a longitudinal fissure. This is that big, deep crack in the middle of your brain. It kind of goes in a sagittal aspect, and this basically separates your two hemispheres, your brain, into right and left hemispheres. Then there's a lateral fissure, and this one is kind of a little further down. This will separate the temporal lobe from your frontal and parietal lobes. And so one of the things to remember is that the regions of your cerebral cortex are divided up into regions that share the same regional names as the skull bones that they're underneath. For instance, your cerebral cortex, the parietal lobe, is just underneath the parietal bones. The temporal lobe is just beneath the temporal bones. And where you typically see the squamous suture, that's about where you would see the lateral fissure. So the fissures kind of approximate roughly where some of these fissures are. So the central sulcus is a big sulcus. It's an easily identifiable one, and it kind of separates the frontal from the parietal lobes. It's basically kind of a coronal aspect. It takes a coronal aspect that cuts your cerebrum into a frontal lobe, so a front and the back, and the parietal lobes would be in the back. And so they're kind of major landmarks, and we can kind of see where they are. So those are some of the main grooves, the sulci and the related structures like fissures. But what about the gyri? Are there specific gyri that basically can give us a landmark? And the answer is yes. Around the central sulcus, you'll see a couple of gyri. One is called the precentral gyrus. That basically means it's in front of the central sulcus. So it's anterior. And this is going to be your primary motor cortex. And so it's in control of motor functions. And then there it is again, right? More motor functions. We have a lot for an organism. We have a lot of motor control and regulation. So a lot of control of ambulation, movement, and things of that nature. A lot of it. The postcentral gyrus is just posterior to the central sulcus, and it's going to be your sensory cortex, your somatosensory cortex. So it's taking in sensory information. Now, around these structures, and so in the middle, between precentral and postcentral, you would see your central sulcus. So the cerebral cortex then is composed of both gray matter, which is typically on the outer regions of the cortex, and white matter, which is a little bit more inside. Now, there's a couple of different types of neurons. Remember, we said there's different types of neurons in the cerebellum, and then there are different types of neurons also in the cerebrum. You have what's called the fusiform, we have the stellate again, so and the pyramidal cells. So we'll take a look at some of these here in just a second. But the first thing we want to do is identify some of the main lobes, and then we'll take a look at a picture of the brain so you guys can see what's going on. So some of the main lobes we have are certainly the frontal lobe, and that's got the region that's just underneath your frontal bone of your skull. And what's happening here, because it's anterior to the central sulcus, where your precentral gyrus is, this is going to be involved in voluntary motor function, aggression, sense of smell is going to be here because that's where your olfactory bulb is. You're going to have mood in this area. Personality comes from this area. Decision making in this is in this area as well. And so this is one of the reasons why if somebody has a frontal lobe injury, a lot of times they can experience either a loss of smell, or they can start to have like aggression problems or personality changes. This is something that's common in somebody who has brain damage in this particular area. The parietal lobe, which is just below the two parietal bones, you have right and left lobe, is going to be behind the central sulcus. So it's in that post-central gyrus area and back. So this is going to be involved in sensory information, both the reception, evaluation, integration of that, except for smell, because smell is up there in the front with your olfactory bulbs. But your hearing and your vision is going to be moving through your parietal lobe. So there's a lot of other sorts of sensory things that are happening. The occipital lobe, which is below the occipital bone, where that's behind or the back of your head. This is going to be really involved in your visual input, so receiving and integrating visual stimulation. The temporal lobe is going to be involved in hearing, and it's also going to evaluate and receive your smell. So your olfactory bulb will basically move through the frontal, that's where it is, and then it'll carry that information to the temporal. And the temporal is a really important lobe. It's kind of an interesting lobe. There's a lot going on there, because not only is this where your hearing region is of your brain, but you also have your memory here, abstract thought, your ability to make abstract thinking, and judgment. So if you have poor judgment in your temporal lobe, it could be to blame. And so these are your main lobes. And there's a lot more, obviously, to these, and it's a fascinating area. Like one, just to give you an idea, just as an example, one example of some of the interesting sorts of things that happens is there was a research study that was done on the temporal lobe of individuals. And so what they wanted to do is they wanted to see what the temporal lobe did with respect to being able to sense that somebody was in the room, right? We've always done this before. You know, we go into a room, we don't really see anybody there, but we kind of sense that somebody's there. And the research basically went something like this. They blindfolded the subjects, and they kind of put like a little helmet on them, and they were able to differentially stimulate the temporal lobe. And they would do a test. So in some cases, they would have somebody in the room with them and see if they could sense if somebody was in the room with them. And other times they would not. And they would do this under different scenarios where they would stimulate or not the temporal lobe. And one really interesting and intriguing study that they found was that when the room was empty and they stimulated the temporal lobe, people expressed feelings of somebody else being in the room with them, which I thought was very, very fascinating and very interesting. So that was like, there's a lot of interesting studies like that with the brain. The insula, that was free, by the way. The insula basically is deep in the lateral fissure, and oftentimes this will take in taste information. And so you can kind of see a lot of your senses are kind of going through this particular area. So here's kind of what it looks like. Here's your brain. You have your two hemispheres, so your right hemisphere on this side and your left hemisphere over here. This main groove in the middle is your longitudinal fissure. So that's where that guy is right there. Your parietal lobe basically is going to be right about where your parietal bone would be. And so here's your parietal lobe, like that. Your central sulcus is going to be, it's usually a little bit of a deeper groove than usual. So there's your central sulcus right there, with your precentral gyrus on one side, your postcentral gyrus on the other. And in some cases, you can actually see it a little bit more. It's a little harder to see in this particular aspect, the superior view. You can see it a little bit better in the lateral view. So for instance, if you turn it around and look at the lateral view, you can see more clearly the central sulcus, which is a deeper sulcus that kind of goes throughout the brain. And it marks the boundary between the frontal lobe, which is in the front underneath the frontal bone, and the parietal lobe, which is just behind it. Then, of course, you can see how you have your lateral fissure, which basically looks like it roughly follows about where you would see the squamous suture, just underneath the temporal bone. And so that forms your temporal lobe. And then your occipital lobe, which basically also follows the lambdoid suture. And that basically is where your visual cortex is. And so you can see the basic landmarks and the basic areas of the brain itself. So when you take a look at the different types of matter, like in the brain, the gray matter versus the white matter, the gray matter is basically divided up into layers, so 1 through 6, and you have your different cells here. For instance, your pyramidal cells, which is one of your neuron types, so called because they kind of look like this little pyramid shape. So this kind of like little pyramid, that's why they're called pyramidal. And so they'll have long axons with little trigs, and then you can see a pseudo unipolar and a couple of bipolar neurons here as well. But you can see different types of neurons in this gray matter region, and then underneath it would be the white matter. So that's just kind of an idea, just to show you like how the different gray matter, white matter is organized, and how there's different layers with different neurons in there. So then, the white matter, typically, when you take a look at the center portion of the cerebrum, we have the cortical region, which is where you find the gray matter, and then the white matter, which is in the cerebral medulla, the center part of the cerebrum itself, different than the medulla oblongata. But typically, the white matter will form these fibers, and they connect the cortex to different nuclei that are supposed to be doing all of the processing. So, for instance, you have different types of fibers. Association fibers are going to be white matter fibers that are going to connect within the same hemisphere. And so you can see those guys in red, right? So you have these like little connections here that are going to be connecting different regions, short associational fibers here within the same area of the brain, within the same hemisphere. Then you have commissural fibers. These are going to basically connect one hemisphere to the other. And this is typically what the corpus callosum is doing. It's basically gluing the two hemispheres together. And these commissural fibers are the blue ones. So you can see you're basically connecting, going from one hemisphere, and you're connecting through that corpus callosum through what's called the the commissure. A commissure is kind of like a little passageway that you can kind of move fibers through, and it connects to the other hemisphere. This is so you can coordinate your two hemispheres and what they're doing. And then the projection fibers are basically going to be going from the cerebrum to the other parts of the brain and the spinal cord. So, for instance, they're going to be going down from one hemisphere through the thalamus, through the spinal cord, and so forth. And notice how they decussate. Right? We talked about that before, where they cross over, and that's where you get this sort of phenomenon where the right side of your brain controls the left side of your body. And so those are the fibers. And so this is an image of, it looks like a dissection where they actually stained for those specific types of fibers themselves. Okay, so what are some of the main nuclei then? We just talked about some of the connections, the associational fibers of connecting different regions of the brain to different nuclei, connecting the cerebrum, and so forth. So what are some of these nuclei and what do they do? Well, the basal nuclei is a big one, right? So this is one that you want to put a star next to, because the basal nuclei is really, really important. Its job mostly is motor function control. You can find it in your cerebrum. We talked about it already in the diencephalon and in the midbrain. In the cerebrum, it's called the corpus striatum, or the striated body. And you have a couple of things in there, a couple of nuclei. So one is called the caudate nucleus. Another one is the lentiform nucleus, which has within it structures called the putamen and the globus pallidus. And so these are regions within that lentiform nucleus, structures associated with that. And then outside of the cerebrum, you have the subthalamic nucleus and the substantia nigra, where we talked a little bit about the substantia nigra and the midbrain. And these are in the subthalamic nucleus we just talked about in the subthalamus. So here you can now see that you have different regions of the brain that are all starting to connect together and sort of starting to work together. And this is kind of what it looks like from a side view. So if you were to take a look at a sagittal view of the brain, you have in the cerebrum the corpus striatum. So the caudate nucleus is kind of like this ram's horn looking structure that terminates into this sort of bulbous region, which is the lentiform nucleus. Together they form the corpus striatum. And then at the tip of this caudate nucleus is going to be your amygdala, where this is the seat of fear and things of that nature. We talked a little bit about the amygdala already. Now, below this structure, this ram's horn-like structure, is going to be your subthalamic nucleus. So if you notice in position, this is about where the thalamus is. So you can see the thalamus is right here, kind of behind this corpus striatum. And then below that, a little more in the midbrain, is your substantia nigra. Now, all of these guys together, the caudate nucleus, the lentiform nucleus, the subthalamic nucleus, the substantia nigra, is basically collectively referred to as the basal nuclei. And that's motor control. So if you take a coronal view, you can see also the way that it's organized. So basically, if you take a look at this, you'll see the caudate nucleus on either side. The lentiform nucleus, which is then composed of the two pieces. So the putamen is the outer region, the globus pallidus is the inner region. And so those are the two pieces of your corpus striatum. So when you take a look at a coronal view, you don't necessarily see this in the detail of this. You have to kind of stain some of these things to be able to see this, but you're actually looking at an aspect where you can see the caudate nucleus and the lentiform nucleus. Okay, more cerebrum and diencephalon, the limbic system. We've talked about the limbic system, right? So the basal nuclei is a big one. Where'd it go? There. So this is a big one. This is one that's worth spending some time on.
and getting to understand well, okay? Because it's kind of like the master executive team of all motor function and control, okay? Now, the limbic system is another one that's worth spending a little bit of time on because this is what really sets us apart, right? Because this is what we refer to loosely as our emotions, our emotion center. And because our emotions are kind of accentuate our experiences, they oftentimes will feed and reinforce our learning and our memory and our motivation. So, why we do the things we do. This is different, right? Because, first of all, your motivation in particular, for most animals, is mostly instinctual. But in us, it's not. So, for instance, when we do something, it's oftentimes driven by our emotions. When an animal does something, it's oftentimes driven by their instinct. And so that creates a lot of more complexity in the human condition. It also accentuates learning. The reason why we are masters at learning is because we link our emotions to our learning. So the more I can make an idea that you're learning emotional, the more likely you are to learn it. Same thing with memory. The more emotional a moment is, the more likely we are to remember it. So we use our emotion to facilitate the things that we do beyond instinct and the things that we learn and remember so that we can actually have the capacity to learn more, to know more, and to do more. This makes all the difference. So, this also will feed and influence our endocrine system and our autonomic nervous systems. So, it also integrates in other parts of our nervous system. So, the limbic system typically has 6 areas, and they're basically connected by nerve tracks, like the fornix, right, which is the lower portion, like the corpus callosum. You have the corpus callosum on the sagittal view of the brain, and then you have the lateral ventricle, and then below that, the bottom floor of it would be the fornix, and then below that, you have the thalamus. And so in the cerebrum, you have the cingulate gyrus and the perihippocampal gyrus. And the hippocampus is important, as well as the amygdala, because the hippocampus typically is associated with your memory. Your amygdala is associated with your motivation and your fear. And so these are all important pieces. So these are 4 of the 6. The subcortical nucleus is an anterior anterior nucleus of the thalamus and the habenula. So this is basically going to feed into your emotions. Right, we already talked about the habenula already, right, as part of your epithalamus. And then the hypothalamus has the mammillary bodies, which we also mentioned was also in contact with your limbic system. And of course, your olfactory cortex, which allows you to make emotional responses to your olfactory reflexes. So now basically, if you kind of go back and you connect all these little bits and pieces that we mentioned, for instance, when we talked about the mammillary body activity in the hypothalamus, and then we talked about the amygdala, which was actually before that, which was earlier in the chapter. And then kind of take these little pieces like the lateral dorsal nucleus of the thalamus and these different areas where we're talking about how it interacts with the limbic system, like for instance, your hypothalamus, these areas, these are all coming together now as a group. Just like the basal nuclei is a collection of all these different nuclei that are in geographically or anatomically different areas, but functionally, they're coming together to form the limbic system. So that's the important thing to understand. So anatomically, these things are in different places. But they functionally act as a team. So it's kind of like a network, right? So you see the same thing with your motor control. There are parts of your brain that are basically kind of spread out in different anatomical locations, whether it's your midbrain or your cerebrum, your thalamus. And so they all come together. They're located in different anatomical regions, but they all come together to act functionally as a team together. And the limbic system is the same way. So this is kind of what it looks like for your limbic system. So your fornix, which is the bottom floor. So you have your corpus callosum up here in this region here, and then you have your lateral ventricle, and then your fornix, which is this region just below that. And now you're going to see you have all these different structures that almost kind of looks like an earpiece that you'd like wear around your ear. But the first thing you're going to see is the anterior nucleus of the thalamus. That's going to be this upper portion, this large piece right here. And then there's going to be your anterior commissure, which is kind of like a little isthmus that basically connects the two halves. The olfactory cortex, which is going to be in this region, and of course your olfactory bulb, which is taking in olfactory information, your sense of smell. Right behind in your hypothalamus, you're going to see your mammillary body, which is actually part of this. You can see it kind of connects together with the anterior nuclei of the thalamus. And then you're going to see a couple of things. First of all, just below your mammillary body is going to be your amygdala. Your amygdala is going to be connected here to your olfactory cortex. And so they're communicating back and forth with each other. That gives you emotional response to your sense of smell. You can see how your mammillary bodies are also connected to the anterior nuclei of the thalamus. And then the habenula, which is part of your epithalamus, is going to be interconnected as well with the anterior nuclei of the thalamus. And your hippocampus, which is basically just behind your amygdala, is where your memory is going to be. And just below that is going to be your parahippocampal gyrus. And so these are all kind of clustered together, and they kind of form together your sense of emotion and learning and memory. So this is one of the reasons why your emotions are deeply involved in your memory and your learning. Okay. Let's pause there for a second. How are you guys doing? You doing okay? That was a lot. And we're in the middle of a topical shift right now. So I just want to just do a temperature shift. You guys are still okay? You're still with me? Then give me a thumbs up. Yeah, there's definitely a ton of information. There's a lot, right? So and the crazy thing is this is just barely scratching the surface of all the things that we've learned about the brain. So this kind of gets us started. Which is one of the reasons why the next exam I only want to have on 2 chapters to give you guys enough bandwidth to be able to dig into them. Okay, so this is a topical shift. We're going to talk about the coverings of the brain, the meninges, the ventricles, and the cerebral spinal fluid. So basically the brain itself is going to be wrapped up in connective tissue layers called the meninges. These are membranes that basically cover the brain, they protect the brain, and you have 3 main layers. The outermost layer is the dura mater, right? So that's the tough connective tissue husk. And some of you guys saw some of that in your sheep brains. Below that is going to be the arachnoid mater, which basically looks like spider webs. That's why they call it arachnoid mater. And then basically on top of the brain itself, bound tightly to the brain, it looks like cellophane, is going to be the pia mater. Now there's also spaces as well. So just like there's 3 layers, right? So going from outer to inner, dura, arachnoid, and pia, there's also spaces. So for instance, the subdural space, which would basically inhabit the area between the dura mater and the arachnoid mater, is basically full of serous fluid. The subarachnoid space, which would be the space that inhabits just below the arachnoid mater and above the pia mater, is basically going to be where the circulating cerebrospinal fluid is inhabited. And so here you can see a cutaway of the brain where you can see your three meningeal layers. So you have your dura, your arachnoid, and then your pia, which is this little cellophane-like structure that's right on top of the brain itself. And so below or above the dura mater is going to be the skull bone. And so the dura mater has two different pieces to it. The lower portion of the dura mater is what's called the meningeal dura, which basically allows it to make contact with the membrane below it. And then there's the periosteal dura, which is basically more connective tissue so that it can integrate with the overlying skull bone. And this is kind of like a cutaway. So here you can see in the anterior median fissure, we can see that the dura, basically the meninges dive down into that fissure and they cover the fissure itself. Okay, so let's start off one layer at a time. So the dura mater, basically we have our two layers. We talked about them, right? The periosteal dura, the inner meningeal dura. By the way, does this sound like a familiar pattern to you? We saw the same pattern in cartilage. We saw the same pattern in bone, right? You have a sort of a cellular or a membranous inner layer, that's the meningeal dura, surrounded by a more connective tissue outer layer, right? Very much like the periosteum, having a cellular layer on the inner portion and a connective tissue layer on the outer portion. Similar pattern. So the dural folds basically will kind of bind up the brain, and they'll basically hold it together. So inside the longitudinal fissure between the two hemispheres is what's referred to as the falx cerebri. So that's the structure of the dura that dips down into that longitudinal fissure. The structure that's between the cerebellum and the cerebrum. So the cerebellum, remember, is kind of like dangling in the back of your brain, but between the bottom portion of your cerebrum, where your occipital lobe is, and the cerebellum is what's called the tentorium cerebelli. So this is going to be a dura mater layer that sort of separates those two. And then between the two cerebellar hemispheres, just like you have between the two cerebral hemispheres, is the falx cerebelli. So this is a dura mater area where the meninges dip down into these deep fissures and they cover the brain so that the brain is completely covered. And so you don't get a lot of, chafing of the cerebellum isn't chafing against the cerebrum. The meninges are intervening and covering and wrapping each area of the brain to insulate it and to protect it. So at where you see these three folds, you basically have what's called venous sinuses, and we'll talk more about that. So a venous sinus is where the dura separates, and it basically drains venous blood and cerebrospinal fluid from the brain. So this is kind of like a vascular structure. That helps to drain blood and CSF from the brain itself. Remember, you have CSF circulating in this area. And so you want to make sure you can drain this and not just have it build up and have pressure build up there. So the arachnoid mater is basically this really thin area. So that's given here by the pink area. So just below the dura mater is going to be your subdural space. And then below the arachnoid mater is going to be the subarachnoid space. And the subarachnoid space, which is kind of the sandwich between, the subdural space rather, which is the sandwich between the dura and the arachnoid. There's a little bit of fluid in here just to kind of lubricate things, to kind of make sure everything is lubricated. You don't have a lot of chafing. But where the main circulation is happening is in the subarachnoid space. So the last layer we have is the thin pia mater, which is basically this little thin piece. It's very thin, very delicate. It sticks to the brain. You typically can't really peel it off of the brain. It's very tight against the brain. It kind of connects the brain. It's like skin tight. And so it's there to protect the tissue, the neural tissue. Now, just above the pia mater and below the arachnoid mater is the subarachnoid space. And this is basically where you're going to have these web-like strands. That's the arachnoid piece, spider webs. You're going to have blood vessels in here, and you're going to be circulating your cerebral spinal fluid area. This is where your main circulatory space is for your cerebral spinal fluid. And we're going to take a look at this here in just a second, as we see where the cerebral spinal fluid circulates around the brain itself. So those are the meninges, right? And they kind of form a little bit of the structure that we need in order to be able to move fluids around the brain. However, there's more areas in the brain for fluid manipulation, and that's the ventricles. So the ventricles are basically open chambers in the brain that help to circulate CSF, cerebrospinal fluid. So generally speaking, they will develop during the neural tube development, like we saw before, and they're lined with a specific type of glial cell called ependymal cells. And so you have 4 different ventricles. You have the lateral ventricles, you have which form the first 2. So each one of these is within the cerebral hemispheres, and they're separated from each other by what's called the septa pellucidum. And so this is connected then to the third ventricle. So if you take a look at the brain in the coronal aspect, you'll see that you have a lateral ventricle like this. You'll see the septum pellucidum, the second ventricle, which is a lateral, and then below it, you'll see like this T shape, which would be like the third ventricle, just below it. Typically, that dips into the diencephalon. Remember, we said that it surrounds the interthalamic adhesion. And then we'll connect to the fourth ventricle by way of the cerebral aqueduct. So the cerebral spinal fluid will circulate from the first and second. With the laterals, it'll go into the third, and then from the third, it'll basically go through the cerebral aqueduct to that sort of triangular shaped fourth ventricle that's smashed between the cerebellum and the brainstem. So this one is basically connected. So it basically drains between the medulla and the spinal cord, or the medulla rather, and the cerebellum. And the important thing is its connection from here will connect to the subarachnoid space. And so there's little apertures or holes called lateral and medial apertures that will allow the CSF to move into the subarachnoid space. And then once it's in that space, it can circulate around the brain. So this is kind of what it looks like. So the ram's horns, the blue ones, this is going to be the first and the second, right? So these are the anterior horns of the lateral ventricles. So the first one is up here. Next to it is the second lateral ventricle. And so they're connected by the interventricular foramen. And then below that, you'll see the connection, the interventricular foramen draining down into the third ventricle. And so this little structure here, that little open spot right there, that's your interthalamic adhesion. So kind of wrapping around that thalamus area, that's where the third ventricle does basically. And then what's going to happen is from the third ventricle, it's going to drain down the cerebral aqueduct, which is this little channel. that drains then into the fourth ventricle. And then the cerebrospinal fluid will then go down through the central canal of the spinal cord. And so that's where your spinal fluid comes from. Your spinal fluid is cerebrospinal fluid that comes from the ventricle system and then goes down into the central canal of your spinal cord. Okay. This is a great place to stop, because now we're going to talk about cerebrospinal fluid, and we want to make sure we have plenty of time for that. Not only that, but we've already got plenty to work with. So on Tuesday, we will pick up and probably pretty easily finish this chapter, and then we'll move into chapter 12 after that.